Hey there, we all think we know milk, right? But what if I told you its journey from the farm to your fridge is full of these hidden transformations? Today, we're going to dive into the secret life of its proteins.
Seriously, that milk you pour on your cereal has been on a wild ride. And the very things we do to make it safe and last longer, well, they completely change it on a molecular level.
Okay, so first things first. Milk isn't just, some simple white liquid. It's actually this incredibly complex biological fluid just packed with all these nutrients designed by nature. So yeah, you can see it's mostly water. But our story, the really interesting part, is hiding in that tiny little 3.5% slice for protein. We're going to zoom way in on just one of those proteins to see what really happens to it on the way to your kitchen. All right, so let's meet the main character of our story. This little guy is the most abundant whey protein you'll find in cow's milk, and its job is super important. It's got a fancy name, Beta-lactoglobulin, but let's just call it BLG. And a great way to think of it is like a tiny, all-natural delivery truck. Its whole purpose is to carry around important stuff like vitamins and fatty acids.
And to do that job, its structure is key. In its natural state, just as it comes, BLG is folded up into this perfect, compact little shape, kind of like a tightly wound ball of yarn. And that perfect fold is what protects the cargo it's carrying inside.
So, the big question is, what happens when this perfectly folded little ball of yarn runs into the gauntlet of modern milk processing? The heat, the pressure, what happens then?
So this milk goes on quite a journey. First, it gets heated up to separate the cream. Then, it's put under insane pressure, that's homogenization, to break up the fat so it doesn't separate later. And then, boom, it's heated again for pasteurization to make it safe to drink. Every single step involves force and, most importantly, heat.
And it's that heat, especially from pasteurization, that really does a number on our little protein. Remember that tightly wound ball of yarn? Well, the heat makes it start to unravel. The scientific term for this is denaturation. Basically, the protein loses its original neat shape, and all its hidden inner bits are suddenly exposed to the world.
And what happens when yarn unravels? It gets messy. Once these proteins are unraveled, they get kind of sticky. Those newly exposed parts of one protein strand start grabbing onto and tangling up with others. This whole clumping process is called aggregation. So you go from these nice individual little yarn balls to one big, messy, tangled knot.
And it doesn't stop there. Something else happens. The natural sugar in milk, lactose, it actually starts to stick to these unraveled protein strands. This is called lactosylation. So now our messy, tangled knot of yarn is also getting coated in sticky sugar, changing it even more.
Okay, so let's recap. We now have these proteins that are unraveled, clumped together, and coated in sugar. They're totally different from how they started. So, how does our body react to this new creation? This is where the story gets really fascinating, especially when we look at digestion and our immune system.
Now, you might think this new shape is harder to digest, but in the stomach, something kind of surprising happens. The original, perfectly folded protein is actually pretty tough. It resists digestion. But the unraveled version, well, it's often easier for our digestive enzymes to attack because all the spots where they need to cut are now out in the open.
But it's not that simple. It's really a mixed bag. While just unraveling the protein can make it easier to digest, those other changes, they can slow things right down. The big clumps can hide the spots where enzymes need to work, and those sugar molecules that got stuck on, they can literally block the enzymes from getting in there and doing their job. Okay, so if the shape is different and how we digest it is different, does our body's security team, I'm talking about the immune system in our gut, does it see these new proteins differently too?
And it absolutely looks like it does. Research shows our body uses different doorways depending on the protein's shape. The original native BLG protein, that gets absorbed by our standard gut cells. But those big, clumpy aggregates that were formed by the heat, they tend to get picked up by these special immune surveillance centers in the gut called Peyer's patches.
And because they're coming in through different doors, they can ring different alarm bells, so to speak. The original protein is linked to what's called a Th1 type response. But the clumped up aggregated protein, that's linked to a Th2 type response, which is the pathway often associated with allergies. It's pretty clear the body is telling them apart and reacting differently.
So after hearing all that, what do you think? Is milk processing our friend or our foe? Well, like with pretty much everything in science, the real answer is, it's complicated. It's a trade-off. And you can see that trade-off right here. On one hand, we process milk for really good reasons: safety, to kill harmful bacteria, and stability, so it lasts longer on the shelf. But the unintended consequence of doing that is changing the natural structure of these proteins, making them unravel, clump up, and get all sugary.
And get this, the temperature where all this starts to happen isn't even that high. We're talking about 65 degrees Celsius or about 150 Fahrenheit. That's a temperature that gets blown past easily during any standard pasteurization process.
So here's the big takeaway. The milk sitting in your fridge is not the same on a deep molecular level as the milk that first came from the cow. The very processes that we rely on to make it safe and convenient for us to drink, will they completely rewrite the identity of its proteins? And that just leaves us with this huge, fascinating question to think about, right? The exact same process that keeps our milk safe also changes it forever, creating something that's really different from what nature originally made. So the question for all of us is, how do we balance that? How do we weigh industrial safety against the product's natural state?